Bueno, muchísimas gracias, María Ángeles, por la bonita introducción. Agradezco. Agradecer a los organizadores el invitarme hoy por estar aquí. Es un placer compartir este ratito. En cuanto al tema, creo que voy a cambiar un poco la dinámica de la mañana. Espero que os sea interesante y que mantenga vuestros niveles de azúcar altos, porque vamos a hablar de digestiones. Y también me habían pedido que hiciera la conferencia en inglés, así que la voy a iniciar en inglés, si no hay ningún problema. Thanks so much. Okay, so let's go to our topic today. Before starting talking about digestion and absorption with in vitro models, I would like to define two concepts that are hot topics nowadays in food research, which one of them is bioaccessibility and the other one is bioavailability. Bioaccessibility, most of us probably will know, like when we talk about bioaccessibility, we talk about the release of nutrients and substances that are in the food matrix, right? But as a hot topic nowadays, there is a discussion on how we best define it. And as an example, I'm bringing this review where it was published this year, where it talks about the need of a consensus. That is something we are going to discuss today a few times. So when we look at bioaccessibility, we can find different situations. When we talk about nutrients, we can have those within a food matrix. This is representing vegetable source, plant source components. So we can have those that are inside the food matrix and can be accessible and hydrolyzed once they are released in the intestinal lumen. Then we can have those that are not accessible and are hydrolyzed once enzymes enter and diffuse through the cell wall. We can have those that are not accessible and could be hydrolyzed as well, and those that are accessible are partially or not hydrolyzed. Then we can have their examples are peptides or anti-nutrients as well. The second concept we're going to define today is the bioavailability. We can say that a compound is bioavailable when the amount of an injected nutrient that is released from the food matrix during digestion is hydrolyzed and then absorbed through the gastrointestinal tract, transported and distributed to the target cells and organs, independent on the case. When we compare bioaccessibility and bioavailability, we have within the scientific community the discussion of using bio when we talk about viability. Because when we talk with meds, they will say, well, that's in vivo data, right? You need humans to do it. So it's now accepted to use the term permeability and transport instead when we talk about in vitro work. And also it's very important to compare the bioaccessibility with the bioavailability or permeability in terms of magnitude, because we can have compounds that are 100% bioaccessible from a food matrix, but then when we talk about the bioavailability, we may have 0 value. So it's not that important how bioaccessible it is when we are talking about, for example, bioactive compounds that we want them to reach, for example, a kidney, let's say. So we might be interested in those that have higher permeability instead. So today I'm going to go through a number of items. So why we need in vitro digestion models, the InfoDigest protocol, for most of you probably is unknown. So I will try to be brief on that, how we treat our samples after digestion and which are the options we have when we work with that type of experiments, how we treat our digesta samples before using them in cell culture models. We are going to see that they are toxic to our cells and we don't want to ruin our experiments and waste our money, right? future perspectives on this protocol, and I'm going to introduce as well the InfoGest network, that I'm part of that international network since almost 10 years, probably, since I moved from Autónoma. This is nothing new. I'm not bringing anything novel in this slide, right? So why we need in vitro digestion models? As any in vitro protocol, we usually choose to run it in vitro because it's more reproducible, less expensive, of course, less labor intense, and very important, is easy to standardize, something we are really focused on it today. Introducing the InfoGest protocol, what we want to do in the lab, in our bench, is to simulate what happens in our body, from the oral phase to the gastric phase and the intestine, large and small intestine. How we mimic that? So we use a beaker or a centrifuge tube, and we mix there our fluids and enzymes to simulate each step of the digestion process. And the more we know about how digestion occurs, the more we know about the health benefits of the food we consume. So starting with the protocol, InfoGest network started in 2010 as a cost action. You might know that cost actions are European projects that they fund network between researchers, and the question that they wanted to answer at that time in 2010 was, can we standardize a protocol to run digestion, right? Because each lab had their own specifics and their own research question to answer. So there was really an interest around the world to answer these questions and to have standardized protocols. Why? Because when we standardize protocols, we can compare results, right? That is very important. The first protocol, it was published in Food and Function Journal in 2014. The list of authors includes researchers from all around the globe. And since then, and once the cost action ended in 2015, the network is still alive nowadays, right? So next steps was a very important one, really a landmark achievement, was the publication of the Nature protocol on the InfoGest 2.0, they call it, version. This protocol is really well defined. You have all the methods, time, incubation, even the reference of the reagents that you need to use. So really, if you ever want to start working on digestion, please have a read of these papers. So basically, I'm taking this figure from the protocol, the paper, so it will give you all the specifics in how to perform your experiments. You need a few days before starting to measure enzyme activity, because previously people were just using, okay, if I need 5, let's say, 1 gram per ml of pepsin. Okay, I buy one here, then another researcher will buy in Chile and another one in Canada. I guarantee the enzyme activity is not the same. You have different brands as well, and then when you want to compare results, it's not easy. So in the protocol, all the enzymes activities, how to measure it, is very well described. But we wanted to move a step further, and what happens at the end of your digestion, you get your samples. You were incubating your samples for 2 hours in the gastric phase, then another 2 hours in the intestinal phase, and then you finish. What do you do with your sample, right? So you have different options, and depending on the research question that you want to answer, you have to choose, right? It's not the same if I'm looking at food structure or I want to run some cellular assays after. But what I was presenting there is the simplest method, which is the static method, where you can use a beaker for each of the steps. And nowadays, there are more advanced modules, not standardized all of them yet. A second version is one which is called semi-dynamic, where you have the gastric phase which is dynamic. That means that the addition of gastric fluids and enzymes occurs during the incubation period. And then the most advanced method is the dynamic in vitro digestion system, but everything occurs in bottles in this case, but are bioreactors as well, because it includes the colon section as well. And that's very important when you want to look at microbiota. And at CL, we have the most advanced model on in vitro digestion, which is this guy here, the SIMG. At this part here, it simulates the stomach, sorry, and this part here we simulate the small intestine and the large intestine as well. So moving back to the static protocol, and I was mentioning that it's very important to know which is your research question, to know which is the next step on your experiment, right? So when you are interested, for example, of looking at the food structure, you can use microscopy, rheology, or even looking at the particle size, right? So just brought an example to put this in context. So let's say we have different heat treatment in our milk, for example, and we dye the protein with green color and the lipid phase with Nile red color. So during the digestion, we have at the initial point, and then the digestion occurs for 60 minutes in the gastric phase, then 120 minutes and 240. Okay, doesn't matter the time. But the important thing we see here, you can see, well, there, it's like the protein is going to disappear because it's digested, of course, and we're going to release peptides. And the lipid phase, it tends to aggregate. So the different structure, it could be seen, actually. How would you treat your samples after you run the digestion? Just keep them on ice, and as soon as possible, run to the microscopy lab and take your pictures. That will be easy, right? But what happens if you're looking at protein digestion? If you're looking at protein digestion, you might be interested on looking at what happened after the stomach phase. So in that case, you can use inhibitors, for example, or you can raise the pH as well, because the gastric digestion occurs with the pepsin. Pepsin is an enzyme that is active at pH 3, right? If we raise the pH, then we are going to inactivate it. Which type of information we can get? We can analyze the protein sequence in our samples and look to the most released peptides. We can have plots similar to this one. And of course, looking at the peptide sequence of the peptide release, which could be related to bioactivity of peptides thereafter. What happens if we are interested on the intestinal digestion and we look also to proteins? We can add also inhibitors that are going to inactivate the activity of trypsin. Trypsin is the enzyme that's going to digest proteins in the intestine. Or we can also, if we can, and it depends on the research question we want to answer, we can heat our samples. And that will be the gold standard and the best option. But imagine you are working on looking at the processing technique, and you are comparing pasteurization with UHC treatment, right? If at the end of the digestion, we treat all the samples at 100 degrees for 5 minutes, it doesn't matter what you have at the starting point, because all samples are going to be the same, exactly. Right, so that will be okay, but if you're looking at food structure, it's not recommended. What if you are looking at carbohydrate digestion? Then you might be interested in starch hydrolysis. So then you are gonna be interested in stopping the activity of that enzyme. That is the one that looks that's gonna digest your carbohydrates. In this case, amylase. Amylase is released with the saliva, and also as well it's been described in the gastric digestion. So you can do is you can raise the pH, adding a base, or you can heat treat your samples, or even using glychloroacetic acid to precipitate your samples, or even ethanol dilution. Sorry. You might be looking to bioactive compounds like polyphenols and carotenoids. And this is also a very hot topic, and many labs are interested in including these phytochemicals into formulation. Let's say, for example, emulsions or liposomes, etc. So in that case, you should be very careful when you run your digestion because those compounds are oxygen sensitive. So you have to prevent the contact with oxygen. How you can do it? You can use argon or nitrogen, and you flash your centrifuge tube, for example, and then you just close it and incubate them for the period that you run into your digestion, usually 2 hours gastric, 2 hours intestinal, in an incubator that I didn't mention before. So you run the whole experiment at 37 degrees. Or you can also prevent the contact with light if they are light sensitive, for example, using amber bottles or just put aluminum foil around your tube. That's simple. And the last example that I wanted to bring is the probiotic survival. You might be looking at probiotic foods, and you run your digestion, you have your final samples, and what you should do if you are looking at survival is just dilute your digestive samples and plate them for bacterial growth. And that's what we do. And we have to choose a good option depending on our interest, right? And the next step in most labs is to look at how those nutrients, bioactives, etc., are absorbed through our intestinal barrier. So in this second part of my presentation, I'm going to show you which is the most known model to do it, that simulates the intestinal barrier, the lumen, and the blood circulation, let's say, well, what passes through. What we want to mimic in the lab is the intestinal barrier, which is similar to what you can see here, but you have a microvilli, and it's, of course, a complex tissue. And in the lab, we are able nowadays to, depending on the option, to run it in a 2-compartment monoculture option, or more complex using a 3-compartment monoculture or even co-culture using a special plates we're going to see now. Then more advanced models use organoids. And just the latest advance, there was a paper published last year on the possibility to have an intestine on a chip that is this big. Amazing, because you can have the full digestion and then the absorption, which is really cool. But it's still, for me, science fiction yet. So what we can do in the lab, and we do it every day nowadays, is running the experiments using special plates called transwells. So these plates, they have a basket, it's an insert, where we seed our cells on top of a membrane that is from plastic material. It could be polycarbonate or polyester. And this membrane has pores of a specific pore size, could be 0.22. So we add our cells, we keep them growing for a number of days. We will see later how we do it. And then on the day of the experiment, we add our digestive samples on the top, on the apical side, and we incubate it usually for 2 to 4 hours or even for a longer period of time to simulate our absorption. And then after that incubation, we get the samples from the basolateral part, that this is going to simulate what? What is simulating this part? This section, this the basolateral fraction. Yeah. Blood, exactly. Okay, but passes to the circulation, a blood circulation, right? So with this part, usually what we could run an analysis, could be HPLC, GCMS, etc., to look at the compounds that passes through. Or we are interested in looking at the health benefits of that fraction as well. And how we can do that? Well, we can expose that media to another set of cells, or we can even grow another set of cells in the basolateral side of our plate. That's another option. And I'm talking about cells, but until now I haven't mentioned them. So the cells that we grow in the membrane are usually CACO2 cells, adenocarcinoma cells. This is how they look under the microscope. And this method is really well known in the pharmacy industry. They use it regularly. And there is a very nice protocol published, Nature protocols, for a number of years. Now it's 2007, well known for the whole scientific community. And a more advanced model includes a culture with AC29 MTX cells. These are also intestinal cells, but they're a bit special. Why? Because they secrete mucus. How they do it? If we expose these cells, AC29 cells, to a drug, methotrexate, it's an anti-cancer drug. And we are talking about cancer cells, so we are going to kill them all, right? But some of them, no. Some of them will survive, they mutate, and those resistant to the drug, they will secrete mucus. That was special. And you may be in the lab working for months until you get a nice culture of those resistant cells. They produce the mucus. That was some years back. And when I was in Canada, 10 years ago, we developed our line ourselves. But nowadays, of course, you can buy from the company. So you don't have to wait to spend 3 or 4 months growing a couple of cells in a flask, because very few will be resistant. So when we culture those two cell lines, and if you are able to use a dye to look at the mucus, you could see something like this. Okay, you won't see anything from the back, of course, nothing, but you could actually look at the mucus. And even if you using particles, for example, and you're interested on how they will react to the mucus, because of course, I didn't mention, but we add this cell line because we want to mimic better what happens in our gut, because our gut has the microvilli, but then on top there is a layer of mucus, right? So that's why this model is more advanced and mimics better what happens in vivo. So this is how the transwell look like. So the well, and it has the insert, and this is the membrane, that plastic material, and that's where we see our cells. And we have this image to show you how it happens. So we add our cells and a certain concentration, and we keep it for how many days? 21 days growing, until we have a fully differentiated monolayer of CAC2 cells, or if we have a co-culture with the co-culture and the mucus layer on top. So yes, this is just to show you that it takes 21 days in the lab to grow them, and it's long period of time. And be careful to don't contaminate your plates, because then you have to start all over again. And then, yeah, PhD students freak out. So I mentioned that after 21 days, we have that fully differentiated monolayer. And how do we know that is okay and it's functioning okay? Well, how we do it? We measure the transepithelial resistance. And how we do it? We have this device here, but it's gonna that it has an electrode, and it passes through an electrical current. And we introduce the electrode in our plate, in our well, and it's gonna give us a value, right? So if we put it on day 0, the value would be 0 there, right? But after 21 days, you will reach around 1,000 ohms per centimeter square. It depends of the clone and the cells that you have. Actually, this is interesting as well. I just mentioned it, that we are running a green trial to standardize this protocol, to run this type of experiments, because it's not standardized yet. And actually, we're doing it this summer, and different labs around the world use different clones of the CACO2 cells. And we can find the values of the tier values from 1,000 to 100 after the 21 days. So just to give you an idea, that is not easy to standardize it. Yeah, and depending on the clone, cell clone. So if we want to compare results, we need to standardize, right? That's a message that I wanted to give. And next, I would like to present this paper that we published last year already. Yeah, that was last year, on recommendations on how you should treat your samples, digestive samples, before to run your cellular assays. I don't know how the time, okay, 42. I will, so we critically review the practices that are usually used nowadays, how to detoxify your samples after digestion. Because after digestion, using the model that I explained, your digestive samples will have enzymes, will have trypsin. That is the enzyme we use routinely when we grow cells. And that enzyme is active unless we add an inhibitor. So one of the options that we're showing you before, right? And that enzyme is the same one that we use to detach cells in the lab. So we have to stop the trypsin activity always before running this type of experiments. Biosalts, we add biosalts in our digestam when we mimic the digestion process, and it can change osmolarity. And osmolarity, the cells, Caco2 cells, are very sensitive to it. So we have to be careful with that. And food-derived compounds that are released during digestion, it can also affect the viability. For example, free fatty acids. So depending on the challenge that you have, which we recommended in the review, is always state the enzyme inhibitors that you use and the concentration in the material and method section of your paper, because then you ended up looking at the paper and they will say, yeah, we use the InfoJS protocol with minor modifications. Okay, which are those? And then we treat cells with blah. Okay, but you don't say which inhibitor you use or the concentration. That's typical of what you find. Temperature. You can also treat your digesta samples, and it's with a high temperature, and is suitable for cell culture. That will be the best option, as we said before, because it's the cheapest as well, right? Inhibitors are very expensive. But you have to be careful because it can cause denaturation, as we mentioned before, or aggregation of proteins, the production of advanced glycation products. If you have heat sensitive compounds, also you will affect them. So you have to be careful with that as well, or even oxidation. pH. Well, just make sure always that your samples are a pH which are physiological, 7.5. Sometimes you need to add an acid to decrease the pH and maintain your compound. Just look at that. And sometimes you also use cell culture media because it contains fetal bovine serum, which is used to, usually when we grow cells, we use media with FBS to stop the trypsin activity when we are passing cells, right? So you can use that with your digesta samples to stop trypsin activity, but just keep in mind that if you dilute a lot your samples, then you have to be careful when you want to detect, for example, with advanced techniques like HPLC or so, because you're going to dilute a lot and you will could be below the detection limit of the equipment that you are using. Physical centrifugation is also commonly used to remove lots, but it happens that you will remove a large portion of macromolecules. You have to be aware of that. Dialysis is also used, and filtration as well. In the review, we included a number of tables and a flowchart to help researchers deciding how to do it. And just briefly, I'm going to go through the future perspective of the InfoJS protocol. What I presented today is the Agile model, but nowadays there is an interest of looking at the infant model and elderly as well, depending on the population group you are interested on, or the type of products that you're working. So just give you an example, if we are looking at the elderly population, the pH is a bit higher in the gastric phase. Of course, the enzyme concentration is going to be reduced, and the period of incubation is going to be longer because the gut is slower in general as well, okay? And within the InfoGest network, it has been published already the standardization of the agile model, as already mentioned, in the static and semi-dynamic models. Also for the infant and the elderly is in progress. The semi-dynamic, the dynamic mod, the static model was already published last year. Just to add an extra difficulty on this, interesting as well to look at the gender difference on digestion, because I'm not an expert on this. I haven't tried different protocols for that, but it's also something of interest in the research community as well. And there are differences on, for example, the protein digestion, and it was published in this paper a couple of years ago. And last but not least, I will say, the InfoGest network, just to present. So nowadays they are interested in different topics, and it has evolved since it started. So from in vitro digestion models to lipid digestion in silico, and I'm part of the absorption modules where we are looking at the standardization of the protocol I was mentioned on CACO2 cells. If you are interested and want to know more about digestion, just presenting, there is a very interesting YouTube channel on InfoGest where you can find videos and conference online during COVID. We had also the InfoGest conference online. And they organize always every 2 months, approximately, seminars, online seminars for PhD students. So if anyone is working on digestion or want to know more about it, please check it out, because it's a great way to learn more about this. Even they have the protocols on how to measure the enzyme activity published. So if any of you is interested. And with this, I would like to thank you all for your attention. And this wanted to present part of the work that I'm now developing at CIAL. Thank you so much.